Hello, I'm going to tell you a little bit about TCS, which is a tool for evaluating multiple sequence alignments. First, a brief introduction. The relatedness of proteins is a very interesting field in the evolutionary biology, as it contains a lot of information about protein families, protein properties, if proteins have similar domains, or if they even have similar functions. Now, something that is very commonly used is multiple sequence alignments, which is aligning multiple sequences, being it nucleic acids sequences or protein sequences, amino acid sequences. Then you align these, or you try to align these in the proper manner and see if they share any relatedness. Now, something that is very important in multiple sequence alignments is the accuracy, which is proportional to your identity, meaning that if you have a higher identity, you will more often have a higher accuracy because it is more easy, it is more easy for your method to properly align your sequences. And if you have a low identity, or if your proteins are more distantly related, you will often get a lower accuracy because it is a lot harder for your multiple sequence aligner to align these in the proper manner. Now, to evaluate these multiple sequence alignments, there are some tools that are designed, and TCS is one of them. Other tools are HOT. HOT estimates reliable fractions in multiple sequence alignments by flipping sequences from left to right when it performs pairwise alignments. Guidance does this through alternative guide trees, by creating alternative guide trees from bootstrap replicates, and the alternative multiple sequence alignments will then be compared with each other, and then you can see what the consistency is of your multiple sequence alignment. Parse does this by removing every sequence in a multiple sequence alignment and then realigning the remaining sequences, and then you can also compare these alternative multiple sequence alignments and then see what the consistency is. And then you also have Core, which is consistency based, and I will talk more further in the presentation about this. You also have tools that filter unreliable positions, like Core is also one of them, gblocks and trimmal. And what they do is they filter out residues or columns that have a low score or are considered unreliable, with which you can use the output for further downstream applications. But eventually, all these tools have the same goal, which is estimating unreliable positions in multiple sequence alignments, and if it is possible in your tool to filter these out and use your output, your filtered output, in further downstream applications like the construction of a phylogenetic tree. Now I'm going to explain the tool with a research question. The research in this case is the research on nerve growths. It is going to be analyzed. So what they did is they're trying to find the relatedness of these 4 nerve growth factors, and they performed a multiple sequence alignment using 3 different methods. They used muscle, MAFT, and K-line, which are very commonly used methods because they are considered fast aligners, but also having a high accuracy. Now, the thing is, they asked which of these methods is the most reliable one, and we are going to use TCS to see if we can quantify this and see if we can find the most reliable method. Now, TCS was first described in an article where they benchmarked TCS to other evaluation tools, and they saw that TCS is a very accurate one. It is even one of the better ones, specifically for improving your phylogenetic tree. And the second article, they describe a web tool or a web server for using TCS as a multiple sequence alignment evaluation tool, and to output your data for phylogenetic reconstruction. Now, what is TCS? TCS is a scoring system, and with the scoring system it is possible to evaluate these multiple sequence alignments, and with the scoring system you can create weighted output or filtered output, and with this filtered or weighted output you can reconstruct phylogenetic trees. It is a tool implemented in the tCoffee suite, meaning that it is an extension of tCoffee, and you can install it locally or you can use the web server. In this example, for the research question, I will use the web server. If you install it locally, you can use it through the command line. Now, what is tCoffee? Because TCS is an extension of tCoffee, I will discuss a little bit about tCoffee. tCoffee is a consistency-based multiple sequence alignment method, and with consistency-based, it is a description of multiple sequence alignment methods or algorithms where they will search for triplets, meaning that they will search for an intermediate that will bind to two sequences. So for example, if you have sequence X and Y, they will search for a sequence Z, where the residue of your sequence Z will bind to the residue of your sequence X. And if it binds to X and it binds to Y, it means that X must also bind to Y. That is the philosophy of consistency based. Now, tCoffee is not only a multiple sequence aligner, it also has a list of other tools that you can use. It does RNA alignment, structure-based alignment. You can modify the output. You can filter the output. There are quite a lot of applications in the suite. And you can install it locally, and you can also use the web server of tCoffee. Now, the multiple sequence alignment is done first of all by creating a library of potential pairs of residues, and it does this through global alignment and local alignment. So you will get two separate libraries, with the global alignment being the most optimal alignment, and the local alignment is the alignments with the highest scores. And then when you have these two libraries, they will assign weights to these pair residues based on the identity of the two sequences in the pairwise alignment, with more identical pairs having a higher weight. And when these are weighted, they will be combined. And if you have any duplicates, the weights of both these pairs will then be added together, giving you pair residues that are more commonly aligned, that these are then weighted higher. Now, eventually you have this library, which is a combination of locally and globally pairwise aligned, and these are weighted, and then eventually there will be another weighting procedure on these pair residues, which is called the library extension, and it is done on the triplet approach, which is based on the consistency-based ideology, which is that if your pair residue, so you have these pair residues, and you will try to find other sequences that can be considered as an intermediate residue. So like I said, you have a residue in sequence X, and it aligns with sequence Y, and you're going to look for intermediate residues where X aligns with Z and Z aligns with Y. It must mean that X will align with Y. Now you have these two pairs of XZ and ZY, and the lowest score of these two pairs will be taken, and then this lowest score will be added to the first primary weight of the pair residue. And then with these weights, you get they will eventually do the progressive alignment. They will create a distance matrix, a guide tree through neighbor joining, and then they will do dynamic programming. But instead of using a Boltzmann matrix, they will use these weights of these pair residues as a scoring system. Now TCS works by defining reliability indices, so we're going to see what the reliability is of residues, of columns, of the entire alignment, and of these residue pairs that are created in this library. So first of all, again, because it is an extension, it is also of the tCoffee, it also first generates a library. You can do this using the tCoffee library, but the default is using a propcons library, which uses a pair hidden Markov model, which is a probabilistic model. What you can also use is fastCoffee, which generates multiple sequence alignments using muffed, muscle, and k-line. And from these multiple sequences that are generated, they are going to take the pairwise alignments from these multiple sequence alignments and use this as a library. Now, first of all, the first score is the pair TCS, and from this score they will derive the residue TCS and the column TCS. So this score, the pair TCS, is the estimation of the TCS score for each of these aligned residues of each of these pair aligned residues of the target multiple sequence alignment. So it is going to try to find all these compatible pairs of residue X with residue Z, residue in sequence X and residue in sequence Z, and do the same for the residue in sequence Y with the residue in sequence Z in this library. And all these, we'll try to find all these pairs, but one pair of these compatible, one of these compatible pairs is called a triplet. And of these compatible pairs, they will take the lowest score, as I mentioned with the tCoffee. And in the pair TCS, they will take the sum of all these triplet scores, of all these lowest scores then, all the sum of these, and they will normalize it by taking the pair scores involving all the residues, involving the residue in X, involving all the residue in X with the intermediate Z, plus the sum of the pair scores of the residues, of all the residues of Y that align with the intermediate residue. Now, as I said, the other values are derived from the pair TCS, so we will go on to the column TCS. The column TCS is an average of pair TCS, so it is the average of the pair TCS scores of all the aligned residue pairs, but in one considered column. And then you have the residue TCS, which is the average of the pair TCS scores of all residue pairs, but it is only involving the considered residue and in the same column. Then you have the sequence TCS, which is derived from the residue TCS score, so it is the average of the residue TCS score over the entire sequence. And you have the alignment TCS score, which is the average of all the residue TCS scores over the entire alignment. So the output of TCS is that you get all these calculated TCS values, and then you can compare these between different multiple sequence alignment. What you can also do is you can filter out the less reliable columns and residues based on a option that you have used in the beginning. So for example, you can say, I want to remove all the columns that have a lower score than 4, or I want to remove all the residues that have a lower score than 4. And with this filtered TCS weighted multiple sequence alignment, you can reconstruct phylogenetic trees. You can also do that. You can also do it with a different manner by using the column TCS score, and where you will do a weighted a weighted output actually, which is that you will multiply your columns proportional to your column TCS score. And a third manner is where you can generate 100 bootstrap replicates, but the 100 bootstrap replicates will be based on also on the column TCS. Now, for the research question, which method gave us the best result? I have inserted all the multiple sequence alignments for MUFT, muscle, and K-line in the TCS, and I will show it to you. So first of all, I will tell you a little bit about the web server. So this is the web server for TCS. As you can see, it is extension of tCoffee. First of all, you can input your file using different formats. I use a FASTA format. You can upload it here by clicking here. You can also show more options. Here you can see for the filtering intensity, if you want to do it based on a column or the residue, what is the minimum, what is the max, and so on and so on. You can even choose which method you want to use for your library computation, and you can also choose which output options you want. So I've already done this for the different multiple sequence alignment methods, and this, for example, this one is MUFT. So again, as you can see, you have your alignment score, your sequence alignments, and your column score. Again, here your column score. But as we can see, there are a lot of gaps. There's a lot of green and yellow. Also quite a lot of blue columns. But if we go more down, you can see that there's a lot more red. So a lot more consistency. The alignment looks a lot better. Now, if we compare this to muscle, which has a slightly higher score, these scores are also slightly higher but also comparable. You can see that here are a lot more gaps. There's a lot more gap introduction. Here is a lot less introduction of gaps. There's also less blue columns, and yeah, it does also have a lot of green here and a lot of yellow, meaning that these are the less reliable positions, and here again are the more reliable positions. So it is quite consistent that the first part is a very difficult part to align for both of these methods, and that the last part is a lot more difficult, is a lot easier to align. So they are more consistent in this region than they are in the beginning. And then if we check at K-line, we can see that it has a lower score than muscle, but a higher score than MUFT. And again, here it is also a little bit lower than the muscle. But here it also has an average sequence alignment for these two neurotrophin. For these neurotrophins. And if you look at the alignment, you can also see a lot more blue columns, also more blue in the alignment itself. Again, it has a lot of difficulties with the first region. Here you have quite some yellow. If you compare this to what they are shifted. No, it's comparable. But in the beginning, again, it has a lot of green. Here it also has a lot of gap introduction. For example, you can see here, it's for every sequence it has a gap except for this neurotrophin. It has a lot of these gaps in columns. So this one looks less reliable than the others. But eventually, if we have to come to a conclusion and say if one of these is the best, then I think we can come to the conclusion that the muscle has given the most accurate result. But what you can also say is that these three alignments, if you look at the TCS scores, they give quite resembling output, except for in the beginning region, where they have a different approach of aligning these regions. Now, so if you look at the TCS scores, eventually what we would tell them, the muscle method was gave the best multiple sequence alignment. But if we look at the TCS scores, they are very similar, and that it is basically the beginning position which gives a lot of difficulties for these three aligners. So eventually TCS is a web tool that evaluates multiple sequence alignments, and it does this using these TCS indexes. And with these TCS indexes, we can change our output, we can weigh our output and try to reconstruct phylogenetic trees. But eventually, if you look at it, you can work around it because it is a consistency-based method. So the method of this program is used by T-Coffee and other consistency-based multiple sequence alignments. So if you want to create a more accurate multiple sequence alignment, it's probably better if you just use one of these consistency-based multiple sequence aligners. But what it is very useful for it is to see which of these positions are reliable and which not. So for example, if we saw in a research question, the more at the end of the sequence, they all gave the same results and the same reliable positions, but in the beginning we could see that the multiple sequence aligners had a lot of difficulties trying to align these trying to align these sequences, and that they all had their own method and they all gave a slightly, well, not really a slightly, but they gave a different result. So that was it. Thank you for listening to me. I hope you thought it was interesting, and good day.